It's a great pleasure today to welcome Professor Phil Hogg, and he's going to work on, tell you something about my favourite organelle, the mitochondria. He's a NHMRC Senior Principal Research Fellow from the Lowy Cancer Research Centre in the University of New South Wales in Sydney. He graduated with a PhD from the University of Queensland. And he's a biochemist, and he explores protein function controlled by a particular chemical modification his team identified. So it involves disulfide bonds, and many proteins have these, but in some proteins they are dynamic. They can form and unform, and they can change the protein shape. So he's developed some small molecules that can target some of these proteins and change that disulfide bond. So, and they can inhibit tumour growth in rodents, and they're in clinical trials at the moment. So today he's going to take you from the start of the story to the end of the story. Thanks.
Thanks very much, Ruth, and it's a pleasure to be here. So, as I understand it, this series has been built around the hallmarks of cancer, which I'm showing here as the most recent rendition of that. And what I'll be focusing on today, talking about today, is the deregulation of cellular energetics as one of the hallmarks of cancer development. So, just to remind you, just the fundamentals of this. When it comes to sort of thinking about metabolism, really at the core of it is the catabolism of glucose, how the cell uses glucose to make the energy and the building products it needs to be able to divide and grow. And core to that is two processes, one that occurs in the cytoplasm known as glycolysis, and the other one that occurs in the mitochondria known as central citric acid cycle, oxidative phosphorylation. But the bottom line is, in terms of the core of it, glucose is catabolized to produce ATP. It gets a few molecules of ATP from glycolysis. It gets a lot of molecules of ATP from oxidative phosphorylation in the mitochondria. But in addition to making ATP, a number of the intermediates of either glycolysis or the citric acid cycle in the mitochondrial matrix are precursors for all the sort of things that the cell needs to be able to do its stuff. Like, glucose 6-phosphate is then converted to various nucleotides, glycolipids, proteins. So all the stuff that the cell really requires comes from, at the beginning, the catabolism of glucose, which is the major nutrient that we get from our diet. So it has been recognised actually a long time ago, 8 decades ago now, that tumour cells or cancer cells metabolize glucose typically in a different way than healthy cells do. And this was recognised by this guy called Otto Warburg. And so what he noticed when he cultured tumour cells, he cultured some cancer cells from the peritoneum, and he noticed that these cancer cells were converting glucose to lactate, and they were doing so even under normal oxygen conditions. So what he noticed is that cancer cells would take glucose and they'd get to pyruvate and they'd convert it to lactate, despite the fact that the cancer cells were grown in a normoxic environment. So they had plenty of oxygen to generate ATP through oxidative phosphorylation, but despite that, they were still making a lot of acid. And this process, he coined the term of known as aerobic glycolysis. So the idea being is that even in the presence of normal oxygen, there seems to be more of a dependence on glycolysis than there is on oxidative phosphorylation to produce the things that the cell needs. And to just sort of try to quantify that in a way, so normal cells, they will, if we just look at where they get their ATP from, normal cells will get about 10% of their ATP from glycolysis and about 90% from oxidative phosphorylation, whereas cancer cells, it's typically around about 50-50, and it'll depend on the type of cancer cell you're looking at and conditions and other sort of things. But in general terms, they sort of split their reliance on ATP reduction about half from glycolysis and the other half from oxidative phosphorylation. So the current understanding of aerobic glycolysis is this, is that the increased biosynthesis required by rapidly proliferating cells is more efficiently supplied by aerobic glycolysis than oxidative phosphorylation. So that appears to be the case. And the reason that is that it appears that the glycolysis intermediates are siphoned out of the pathway and into alternate pathways to supply the anabolism of DNA, lipids, and proteins. So as a general sort of description of aerobic glycolysis, this is why it's thought that cancer cells do this, is that they rely on the intermediates of glycolysis to produce the nucleotides, the DNA, and the lipids. In terms of their requirements, this is probably more important for their ability to sort of, to be able to rapidly divide, etc., rather than relying on the ATP produced by oxidative phosphorylation. Now, it's important, that's not to say that they rely on mitochondria any less, it's just that their shift in terms of where they siphon off the intermediates tends to be favoured more here than here. Now, when we're talking about metabolism, it'd have to be the most involved and most complex system that we have going on in our cells. In that, when you're talking about sort of catabolism of glucose, it really ties in pretty much to every cellular process that we have going on. So it's a complex network, which is sort of illustrated here. So there's glycolysis, there's the oxidative phosphorylation going on the mitochondria, and these enzymes and these intermediates, they're siphoned off and they're used in different ways. So it's a very involved network of interactions, lots of feedback, lots of allosteric control. In general terms, so where a lot of the interest now and the focus is trying to understand, which metabolic enzymes that tumours use, what has changed. So what are the sort of key elements of metabolism that tumour cells will affect? So for example, what enzymes are being expressed in different ways? How is their activity controlled in different ways in cancer cells versus normal cells? Now, our particular interest, which is what I'll talk about today, involves a protein that sort of resides somewhere here in this metabolism network. Now, we got interested in that through our fundamental interest in how proteins work. So what I'll do now is change track a bit and tell you about that, and then that will lead back to our interest in this aspect of tumour metabolism. So, it's actually unusual for a protein to work without being chemically modified in some way. So when proteins are made, they are chemically modified in different ways, which I'll tell you about in a minute, to control how, when, and where they function. So if you look at what we know about biology, it's actually unusual for a protein to work without being chemically modified in some way to control how it works. Now, there are three fundamental types of chemical modification of proteins. What I'll call type 1 is covalent modification of a side chain. So a number of small molecules like, say, oxygen, acetyl groups, and a number of large molecules like ubiquitin and other sorts of things can covalently modify the side chains of certain amino acids and in turn control how the protein works. Actually, 15 of our 20 common amino acids can be chemically modified in some way through these type 1 covalent modifications. And in terms of us, phosphorylation of tyrosine is the most common post-translational modification in our particular cells. So this is certainly the most common sort of post-translational modification. The type 2 involved the peptide bonds in proteins. So as you know, certain peptide bonds in proteins that control how they work. So blood coagulation, complement activation, the apoptosis cascade with the cysteine proteases. All this is an example of a post-translational modification that involves cleavage of certain peptide bonds in proteins. Some peptide bonds can also isomerize, particularly the peptide bonds on the N-terminal side of proline residues. They can switch between the cis and the trans configuration, and this is in turn catalyzed by a certain class of enzymes. And the third modification, which is the one that we're interested in, involves the cleavage of some disulfide bonds in proteins. So conceptually, it's the same idea. As some peptide bonds are cleaved in proteins, some of their disulfide bonds are also cleaved in order to control how they work. And we've called these bonds allosteric disulfides to really distinguish them from the major type of disulfide, which is really a structural one. So we identified the first examples of allosteric disulfides, or this mechanism of protein control, back in the mid to late 90s in a protein called thrombospondin 1 and plasminogen, which is somewhere around here. There we go. So there's now around about 30 to 35, I think, well-established examples of this mechanism of control, or well-established examples of allosteric disulfides, and I've just tried to summarize them here. So what I've grouped these examples in terms of the consequence for the function of the protein. So in this case, this is a situation where cleavage of the allosteric disulfide results in a change in ligand binding. In this case, cleavage of an allosteric disulfide results in a change in substrate hydrolysis, so these are all enzymes. In this case, where your cleavage of the allosteric disulfide leads to a change in proteolysis, so you have a situation where an allosteric disulfide bond is cleaved in a protein, that changes the conformation of the protein, which opens it up to proteolysis by certain proteases. And the fourth type is where cleavage of an allosteric disulfide bond is a key to sort of triggering oligomerization or dimerization, oligomerization of the protein. Now, these are the best established examples. So in each case, we know the disulfide bond that's actually being manipulated. We have a crystal structure. There's at least one crystal structure of all these proteins. We have a good idea as to how the bond is cleaved, and we also have a good idea as to the functional consequences of cleavage of that particular disulfide bond. And so we have this. So this is I think our current list of our established, and there's many more than this that at a more preliminary stage of characterization, such that so we know this is happening in the protein, but we haven't nailed down the disulfide bond that's cleaved, or we do have a structure, but we don't know the functional consequence, for example. So one of our primary research aims over the last 5 to 10 years is trying to find a way to identify these disulfide bonds, these allosteric disulfide bonds in proteins. So let me give you an analogy. So when it comes to cleavage of a peptide bond in a protein, for example, this is just our peptide here that might be cleaved. This peptide might be cleaved by a trypsin-like enzyme, if you like, say thrombin or something else like that. The cleavage of certain peptide bonds in the proteins is largely governed by the arrangement of the amino acids around the peptide bond that is cleaved. Okay, so you have what's known as the P2, P1, P1 prime, and P2 amino acids. So the proteases that are cleaving these peptide bonds recognize this sequence and in turn cleave the peptide bond. And this has been important in terms of how these peptide bonds, how they've evolved, and how the proteases that cleave them have evolved. So we worked on the assumption that there may be something about these allosteric disulfide bonds which is key to how they work. If we could figure out what that is, it'll help us identify these bonds in proteins. And we were sort of assuming the same by analogy again, is that there must be something about these bonds in terms of that's driven their evolution. Now, if we look at how proteins have evolved, typically there's a reason. There's some feature of whatever characteristic of that protein that's really helped drive the evolution of these particular disulfide bonds. So what we did notice, the first thing we looked at to see whether there was any sort of particular sequence or arrangement of amino acids around the cysteine residues that were linking our disulfide bond, but we haven't found anything. But what we did notice was that there was a relationship between the actual configuration or the conformation of the disulfide bond and whether it is allosteric. So first of all, we needed to come up with a way of classifying disulfide bonds. And we looked at different ways of trying to do this, but the one that's really worked for us was based on the 5 angles that make up your disulfide bond. So here's your disulfide bond. Here's your 2 cysteine residues. This is your sulfur-sulfur bond here. And a cysteine residue, this is an example of, so this disulfide bond is defined by 5 what are known as dihedral angles. Okay, so this is really the angle of rotation around these 5 bonds that make up your cysteine or your disulfide bond. And so what we did is if we just consider whether just the sign of each particular angle, whether it's a positive or negative, if you do that, you've got 5 angles, you can come up with 20 possible disulfide bond configurations based on the sign of these 5 dihedral angles, okay? So those in the audience that are good with probability theory probably think, well, there's probably more than 20. There are. So what we then do is we eliminate those ones that are symmetrical. Okay, so we end up with 20 different disulfide bond configurations based on the signs of their 5 dihedral angles. And the 20 different configurations are shown around here. Okay, so these are these examples of these disulfide bonds, all from crystal structures that have been orientated with respect to the sulfur-sulfur bond angle here, the disulfide bond. So if we look at these 20 configurations, you find examples of them, all 20 configurations in the protein data bank. So this is the distribution of the 20 configurations in a bit over 16,000 disulfide bonds in non-redundant X-ray structures currently in the PDB. And you can see that all 20 different configurations are represented in proteins. This is our terminology for these 20 different configurations here. And the one that obviously stands out here in terms of looking at is the most common one is the minus left-handed spiral. It actually accounts for more than, say, a quarter of all disulfide bonds. So this is probably almost certainly, this is your typical structural disulfide bond. It has very low energy. And it's typically a very stable disulfide bond, and so that would probably your sort of go-to disulfide configuration when you're wanting to sort of create a structural disulfide that's there to help hold the protein together while it works. So if we look at just the allosteric configurations, there are 3 that are coming out as being allosteric: the minus right-handed staple, the minus left-handed hook, and the minus positive right-handed hook. So if we go back to that table, as I mentioned, we have 30 to 35 established examples. We know the configuration of those allosteric disulfide bonds. If you look at that table, we have around about 17 examples of minus right-handed staples, and we've got half a dozen examples of the other 2 types. So this was encouraging. So we've got 20 different disulfide bond configurations. Of the known allosteric ones, they're falling into a pattern. There are 3 configurations that sort of coming out of this as being allosteric, and this is what we were hoping to find, some signature, if you like, that will help us to say, look at a protein and decide whether it might have an allosteric disulfide or it might be regulated by that sort of mechanism. And we're beginning to understand why these particular configurations are allosteric as compared to the other 17 types. First of all, in particularly the minus right-handed staples and the minus positive right-handed hooks, these 2 bond types, these 2 configurations carry a pre-stress. They're actually more stressed. They carry a potential energy in comparison to the other 18 disulfide bond types. So let me just describe this. So we recognise this in a rudimentary way, but I'll just describe the data of a recent study from a group at Heidelberg, Sue and Greta, who I think did a terrific study. What they did is they took an unbiased look at the potential energy or the pre-stress on all 20 disulfide bond configurations. So they did molecular dynamic simulations on 667 protein containing all the 20 disulfide bond types. And what they found was that of the 20 types, 2 of them jumped out as carrying what they termed a pre-stress. So these disulfide bond configurations can carry this sort of natural stress. They have this potential energy, if you like, across the bond. The idea being is that they're primed to be cleaved. So if you've got a bond there that's naturally stressed, it's going to take less to cleave that disulfide bond than it is a disulfide that's very, very stable. Okay? And so 2 of the 20 jumped out as being stressed, and they were the 2 of the allosteric ones, the minus right-handed staples and the minus positive right-handed hooks. In terms of where that stress is carried, what they noticed was that the actual distance between the sulfur atoms of the disulfide bond were longer than normal. And the angle between the angle that the disulfide bond, the sulfur-sulfur bond angle, makes with the beta carbon of the cysteine residue was also different in terms of the 20 different configurations. So there's the sulfur-sulfur bond angle, the two that are jumping out from the normal, and this is the angle that the disulfide makes with the beta carbon. It's also unusual. So what they concluded was that it's these two features of these particular configurations that are lending the disulfide a particular stress, okay, what they call a pre-stress. And that's almost certainly what's facilitating their cleavage. Okay, so the idea that of these 20 configurations throughout evolution, they've been picked out because they are primed to be cleaved. You know, they're there to sort of act as a functional switch in the protein. They're not there to act as a strut or a structural bond. So the question you're probably thinking is, but what about the third one? So the minus left-handed hook didn't come out of that analysis as being particularly strained or having any particular potential energy, but there is something about this bond that gives us some sense or some clue of why it's coming up as an allosteric configuration. And that comes back to how it's cleaved. So there are two mechanisms, two ways that allosteric disulfide bonds can be cleaved: by enzymes known as oxidoreductases. So here you have a situation, the grey blob is our protein containing our allosteric disulfide. So these are enzymes that contain a reactive dithiol in their active site, and the cysteine thiolate will come along and attack one of the sulfur atoms of the disulfide bond. You end up with an intermediate mixed disulfide, which is then resolved by the other cysteine thiolate in the enzyme. So you end up with a cleaved allosteric disulfide bond and oxidoreductase. So that's one way the bond can be cleaved. The other way that we know that we've found to be cleaved is through a sort of a thiol disulfide exchange. So in this situation, you have an allosteric disulfide bond in a protein. You have a nearby cysteine thiolate, which is effectively unreactive until the cysteine thiolate is brought in line with the disulfide bond through, say, a ligand binding or a shear stress. For example, proteins that are exposed to shear stress in our circulation leads to a conformational change, brings the cysteine sulfur atom in line, which then results in a thiol disulfide exchange and cleavage of allosteric disulfide. So you're probably wondering what this blue thing is here. This just sort of highlights it's the chemistry about how these bonds are cleaved. So cleavage of a disulfide bond is highly directional. So it occurs, if the chemists in the audience, via an SN2 or a biomolecular nucleophilic substitution type 2 reaction. All that means at the end of the day is that there's three sulfur atoms involved in the cleavage, that is the sulfur ion nucleophile and the two sulfur atoms of the disulfide bond. They effectively have to line up at 180-degree angles for this to happen. So they really need to be sitting on a plane for this chemistry to happen. And once that happens, it happens effectively instantaneously. So, for instance, this is a situation where you have the cysteine thiolate that's just not in line with the disulfide, and the conformational change helps it in line, and that enables that to happen. So it's interesting that of the half a dozen examples of the minus left-handed hooks we have in proteins, all of them bar one, I think, but I'm not certain about that, are cleaved by a thiol disulfide exchange, whereas the other allosteric disulfide bonds appear to be cleaved by oxidoreductases. So look, the numbers are still small, and I'm sure it'll transpire that this won't be necessarily a hard and fast rule. But with what we know now, this appears to be why the minus left-handed hooks are popping up, because they seem to be cleaved in a particular way, in contrast to the other two allosteric disulfides. So if you're interested, if you have a protein that you think might have an allosteric disulfide bond, if you're working in a system that you think might be regulated in this way, we've established this simple online tool which will, if you have a structure of a protein in PDB format, you stick it into this and it'll really, I think, give you all the measures of the disulfide bond that we can effectively think of. It's very easy to use. So, you know, if you think, you know, a protein that you're working with might be regulated in this way, this is a way of sort of giving you get a number of parameters of the disulfide bond, which is a sort of a start. The one thing I will say is obviously it's not a foolproof, this sort of bioinformatics mean. What we do know is that the disulfide bonds in proteins, they can change configuration. So just as a protein in solution will change configuration, it'll move around, it'll adopt particular different configurations, disulfide bonds can do the same thing. So in this case, we have one of the allosteric disulfides in the HIV envelope glycoprotein. Like, for example, we've got, I think, 8. What have we got there? Yeah, there's 8. We've got 8 X-ray structures of this disulfide bond, and about half of them exist in a minus left-handed hook configuration, and the other half exist in a minus right-handed staple. So this is the same disulfide bond existing in 2 different configurations in the protein. And so almost certainly I think is happening in this situation is that it may be, for instance, that the bond needs to switch configuration to the minus right-handed staple before it's cleaved. It's possible it goes back the other way. I don't know. This is just a hypothesis. So it's important to keep in mind. So when you look at the features of your disulfide bonds, you're using crystal structures, which is a snapshot of one particular configuration of the protein. So just always keep in mind that you might be interested in a disulfide. It doesn't have your typical one of those 3 allosteric configurations. It's not to say that it could adopt that. Okay, and so there are things to look for, other things to look for as well, which I'm happy to chat about later. So just keep that in mind. It's one, when you use this sort of bioinformatics. Okay. So that's one way we've been trying to identify these allosteric disulfides, and another way is through a chemical means, and this is what it's led to our interest in tumour metabolism. So let me take you through this here. So what we thought we'd try to do is to find chemistries or chemical reactivities that will enable us to identify the reduced form of an allosteric disulfide in a protein. So here's our protein, here's our allosteric disulfide bond. It can be reduced. In some cases it might be oxidized. And so we were looking for chemistries that will identify these reduced thiols, but only these reduced thiols. So we're interested in a chemistry that will latch on to closely spaced cysteine thiols, and so we were hoping that we might be able to identify the reduced form of an allosteric disulfide bond by using that chemistry. So we looked for particular chemistries, and the one that sort of appealed to us was the chemistry of the metalloids. So in particular, antimony has the highest affinity. So when antimony is in a trivalent state, so antimony 3, if it comes across 2 closely spaced cysteine thiols in a protein, and they need to be typically within 5 to 7 angstroms, it forms these very stable cyclic dithioarsenite structures. Okay, so these are effectively irreversible in a biological sense, and they really are they're very selective for the dithiol, is that they really don't have their affinity for monothiols is well into the millimolar range. Okay, so we have this nice selective chemistry, and you've got this metalloid that will latch onto a closely spaced dithiol and effectively won't come off, but it won't touch single thiols in proteins, of which there's plenty of them, which you're not interested typically. So we were hoping to use antimony, but antimony, there are very few sort of chemical building blocks containing antimony, so that it was impractical really to use it. So we look at the next best option in the periodic table was trivalent arsenic, the atom trivalent arsenic. So just to describe that chemistry, it's the same for antimony. So here's your situation. You have a reduced allosteric disulfide bond. The trivalent arsenic is cross-linking the closely spaced cysteine thiol, and this is forming what's known as a cyclic dithioarsenate complex, and it's effectively irreversible in a biological setting anyhow. And so what we did know was with antimony and arsenic compounds, if they're lipid soluble, if they will just dissolve in lipid, they're really quite toxic to cells and to the body. So we wanted to make a trivalent arsenical that was very water soluble, very hydrophilic. We didn't want it to dissolve in lipid. So one of the compounds we've made, in which we've progressed, and which is what I'll talk about now is this organoarsenical we call PenAO. So effectively it's a cysteine mimetic linked to a trivalent arsenical. Okay, so it's an organoarsenical. It's very water soluble, and so this is the compound we made in an effort to identify these allosteric disulfide bonds. And it's been useful to some extent, but I won't talk so much more about how we've used it for different allosterics. So I'll just really talk about what we noticed when we started using this compound was that, first of all, it actually entered cells quite rapidly. So this is a convenient compound to look where it goes. We can measure arsenic atoms using a plasma mass spectrometry. It's a very sensitive way to measure arsenic atoms in any biological fluid. And so we can measure, for example, uptake of PNAO into cultured tumour cells here. So we recognised it was getting in quite fast. And it was also inhibiting their proliferation. So this is the effect of PNAO on the proliferation of, this is a bank of primary glioblastoma cell lines, and also normal astrocytes. And so this molecule, for reasons we didn't understand, it was actually very good at inhibiting their proliferation with IC50s in the low micromolar or submicromolar range. And it certainly had selectivity for tumour cells compared to normal cells. And if we look at this we look at, other sorts of matched cell lines and cell types, we effectively see the same sort of thing. So for whatever reason, we'd made a compound that was good at inhibiting cell proliferation. So then we embarked on this long journey of trying to figure out how it works. So let me describe about 10 years of work before we really figured out how this was working. And it turns out that what it is a mitochondrial toxin. And so let me just take you through the data that led us to that. So what we noticed is when we incubated this compound with cells, the first thing we saw was an increase in mitochondrial production of reactive oxygen species. Okay, so we're seeing reactive oxygen species being produced from the mitochondria at concentrations of PNAO that were triggering proliferation arrest of the cells. This was also coincident with an increase in acid production in the cells, and also a decrease in oxidative phosphorylation in the cells. So two pieces of evidence to suggest that we're targeting the mitochondria in the cells, these reactive oxygen species, which is really the main source of reactive oxygen species is the cells. When you perturb the electron transport chain, you end up with leakage of more reactive oxygen species into the cytosol. We also found that these effects were coincident with loss of mitochondrial transmembrane potential. So this is a way of measuring the potential of the mitochondrial membranes. So these cellular effects were also coincident with that. And also what we were triggering in the cells was a mitochondrial-mediated apoptotic cell death, as evidenced by activation of caspase 9, then 3. Okay, so for all we can tell, the functional consequence was perturbation of mitochondrial function in the cells. So we then started to look at the mitochondria to figure out what its target was. And so what we did is we isolated mitochondria from cells, we biotinylated our PNAO, and we looked for the major binding partner in the mitochondria. And the major protein that it interacts with is this transport protein, and it is the adenine nucleotide translocase. So let me tell you about this. So this is our mitochondria. This is our outer membrane and our inner membrane, which, as you know, is a bigger surface area. This is our electron transport chain. This is ATP synthase. So the pyruvate that's produced from glycolysis gets into the matrix, shuttled through the TCA cycle to produce NADH, which gives up its electrons to the electron transport chain, which create a proton gradient in the intermembrane space, which drives ATP synthase to convert ADP to ATP. Then the ATP is shuttled out into the cytosol of the cell, where it's then used by a whole bunch of processes via this transport protein known as the adenine nucleotide translocase. Okay, so it's a transporter. It's actually the most abundant protein on the inner mitochondrial membrane. You try to estimate its concentration in there, it'd be in the millimolar range. So it's a very abundant protein on the inner mitochondrial membrane, and it forms a complex with the voltage-dependent anion channel and also hexokinase 2, which is the first enzyme of glycolysis. So it's the enzyme that converts glucose to glucose 6-phosphate, and I'll come back to that enzyme a bit later on. Okay. So for all we could tell, this was our major target. And it sort of made sense with what was known about the transporter. It was known that ANT exists in both oxidized and reduced configurations in the inner mitochondrial membrane, and this seems to change with respect to the proliferation rate of the cell. So mitochondrial matrix thyrodoxin appears to reduce a disulfide bond in an ANT, in which it appears to control its function in ways we don't understand. There is a crystal structure of the reduced form of the transporter, which I've just shown here. So what we then did is we expressed the human protein in a yeast model system, and using mutagenesis, defined the cysteine residues that our molecule was cross-linking. And so there are 3 cysteine residues on the matrix-facing side of A and T, and pan-AO, our trivalent arsenical pan-AO, are cross-linking cysteines 57 and 257. So with what we know from our own work and others, this is where pan-AO is affecting mitochondrial function, in that when cells are growth quiescent, it seems that the transport exists in largely an oxidized configuration. When they're starting to proliferate, you make more ATP, the activity of a number of the glycolytic enzymes and other things are changed, and ANT is no exception to this. And it appears that that mechanism of change in activity involves cleavage of this disulfide bond in ANT via thyrodoxin, and it's the reduced form of the transporter that's then complexing with our trivalent arsenical moiety of pan-AO and effectively, messing with its activity in again ways we don't really understand. We're trying for a crystal structure of this, but we're not there yet. Anyhow, with everything we know, this seems to be the functional consequence for the cell. So this molecule is very well tolerated in mice, and it had some nice anti-tumour efficacy. This is just one of our anti-tumour efficacy models. So at very low concentrations, administration of 1 milligram per kilogram per day pan-AO, we were seeing good inhibition of tumour growth in mice in various sorts of models. So we thought that, we'll see if we can take this into clinical testing. So the first thing we need to do is we need to be able to make the compound, and we need to be able to show that it was stable. And we sort of got lucky in that it's a simple molecule to make. It's a 3-step chemical procedure, and the final product, pan-AO, particularly when it's formulated, is very, very stable. We have a shelf life of at least 2 years. So the material was produced to TGA and FDA standards by the Dr. Reddy's Laboratories, which is an Indian pharmaceutical company. So they made about 0.5 kilogram of this molecule and formulated it for the phase 1 trial. So that was the first hurdle passed. We could make this compound quite easily, and it was stable. So then we embarked on some we knew from all our laboratory experiments it really was very well tolerated, this compound at doses really way above efficacious doses in the mice, but we needed to sort of demonstrate this formally. And this was work done by RDT, now in vivo farm here in Adelaide. So the toxicity was defined in terms of 2 5-day cycles with 2-day treatment period between the cycles. So maximum tolerated doses of 20 mg per kg were found in the mouse and 5 mg per kg in the rat with this administration. The major target organ for pan-AO toxicity was the kidney. But importantly, when you stop administration to the mice or the rats, the kidney repaired itself. So it wasn't an irreversible damage. But anyhow, we'd established these MTDs in mice and rats, and we knew that the kidney was something we needed to look at very closely in the patients, and obviously that is what's being done. So here's the trial design. It's a dose escalation. It's a phase 1 dose escalation study, 3 centres. It's patients with solid tumours, refractory to standard therapy. So these are patients, all types of solid tumours that aren't responding to the current standard of care. And pan-AO is delivered by IV pumps for 2, 4, 7, 14 and 21-day cycles. So when we know with our animal studies, we saw the best efficacy when this molecule was administered chronically. Okay, so when we put it in sub-Q pumps, that's where we saw our efficacy. We saw, in terms of decreasing efficacy, if then we went to sub-Q, IP, PO. Okay, and I'm sure it just comes back to the pharmacokinetics of the compound. We know it's cleared very rapidly in the blood, has a half-life of around about 20 minutes. So with chronic administration, this gives us our best chance of success. So it's effectively administered in the same way, say, 5-FU is for the treatment of colorectal cancer, if you're familiar with that. It's reasonably gentle on patients because they can largely be treated in an outpatient setting. They have a pump that's placed in an IV line, and so the pumps are filled up and they'll last for a week or 2, and they can go home and come back in for monitoring, say, a week later. The main pharmacodynamic measure, so this is what I mean by this is a measure trying to see whether the drug's sort of hitting its target, is FDG PET. So this is a measure of really glucose phosphorylation in tumours. It's not perfect, but really is the best PD measure that clinically that we have for, say, measuring, say, tumour metabolism. And it's, as I mentioned, it's 3 sites. It's the principal investigators, Anne Hamilton and Danny Rishin here at the Peter Mac, Jayesh Desai at Royal Melbourne Hospital, and Lisa Horvath at the Chris O'Brien Lifehouse in Sydney. So this was the first patient that received Peneo here at Royal Melbourne Hospital. This is Jayesh Desai. So far so good. We've treated 25 patients. We haven't seen any dose-limiting toxicities. We're at blood levels of around about 2 micromolar now. We know from all our animal studies we need to get to blood levels of about 3 to 5 micromolar before we start seeing anti-tumour efficacy. And so hopefully we'll be there by the end of the year. I think we need at least another probably 1 or 2 dose cohorts before we're there. But so far so good. It's well tolerated, and we're sort of getting to the pointy end of the phase 1 trial. So in the meantime, you know, if Peneo does make it into the clinic, it certainly won't be used on its own. As almost all cancer drugs now, they're used in combinations. Okay, so what we've tried to do while the trial has been ongoing is, in a rational way, try to decide what would be the best combinatorial efficacy for this particular molecule. What would be the best other good cancer drug that we can combine it with that might give us a better outcome than the molecule alone. And so we tried to do this rationally. So the idea is that we know that Peneo is targeting A and T of this complex. So we thought that, okay, let's see if we can find inhibitors of hexokinase 2 of VDAC. There really are no good inhibitors of VDAC, and no particularly good small molecules of inhibitors of hexokinase 2. But in recent studies, it's been found that, from this group here, found that hexokinase 2 activity is actually regulated by mTOR, particularly complex 1, mTORC1. So mTORC1 controls HK2 expression as well as the expression of a number of other glycolytic enzymes. But in addition to that, it also binds to hexokinase 2 and regulates its activity. So we know that that interaction is regulated by the product of hexokinase 2 function, glucose 6-phosphate. I'll go back 1 second. So hexokinase 2, as I mentioned, it's the first step in glycolysis. First enzyme in glycolysis converts glucose to glucose 6-phosphate. Humans make 4 isoforms of hexokinase 2, but transform cells or cancer cells typically use hexokinase 2. There's a strong association between hexokinase 2 expression and poor survival in patients with solid tumours. And we're just really beginning to understand why they use hexokinase 2 and not the other hexokinase, and this seems to be an important aspect of it. So it works both in the cytosol and also bound to VDAC on the outer mitochondrial membrane. Okay, so it works in both places. Its binding to VDAC is regulated by phosphorylation of a threonine residue in hexokinase 2 by AKT. And it's also bound. The enzyme binds to mTORC1, and that affects its activity. This interaction is in turn controlled by glucose 6-phosphate, which is one of the products of this reaction, in ways that aren't really understood. But anyhow, so here we had a control of hexokinase 2, and we did have good inhibitors of mTORC1. So they're the ones that we decided to investigate, particularly the rapalogs. So when we combine Peneo with one of the rapalogs, in this case it's temsirolimus, we see really nice combinatorial efficacy. So this is effects on, this is a primary glioblastoma line looking at cell growth. This is combinations of multiples of IC50 for either Peneo or temsirolimus alone. And when we combine them at those concentrations, we see, almost complete inhibition of cell growth at concentrations where individually they had very little effect. So we see this very nice combinatorial efficacy, which is great. Now, almost certainly there's more going on here than the hexokinase 2 story, and why I think that is from this experiment is that when we combine both compounds, we are effectively losing the protein from the cell. So it appears to be being targeted at degradation. So this is just a Western blot for mTOR and other proteins in the pathway, AK2, or for EBP1 and GAPDH as a loading control. So you see, with the combination, we're just losing the protein, where it's not affecting other proteins in the pathway. So it appears that for some reason the combination is causing mTOR to be degraded in the cell, and we're trying to understand why that is. So I'm sure that this combinatorial efficacy we're seeing is more than just mediated via hexokinase 2, and we're trying to understand. I could talk about some ideas of why that might be the case. So this seems to be a good combinatorial efficacy. We know it sort of works well in the mice. It's just an example where we've established large tumours in mice, and then we've treated them with the combination, and we see sort of a big increase in the necrotic areas in the tumours. So this combination is really sort of cytotoxic for the tumour cells, and from all we know, it's also very well tolerated. So we're trying to understand this mechanism. I think this, from all we know, is the best combination. So in terms of the clinical development, what we're trying to do now is to get to a maximum tolerated dose in the phase 1 all-comers solid tumours trials, and then to do a small phase 1 extension study with a combination of PenAO and one of the rapalogs. The advantage of these are available in Australia. We can buy them. They're well tolerated. We're hoping, and from what we know, the combination with PenAO is also well tolerated. And so that's where we're trying to get with the clinical development, and that really will tell us whether this compound has any legs or not. At the end of the day, we need to see 1 or 2 partial responses in our patients for us to attract the sort of investment in the development to take it further. And yeah, this is really what we've pinned our hopes on, if you like. So I'll finish there, acknowledge the people involved in the work I talked about today, particularly the bioinformatics that I talked about earlier regarding the allosteric disulphides. Jason Wong and Brian Schmidt were key to some of that work. Pierre Dilder has led PenAO's development, the preclinical development of PenAO in my lab. Stephanie D. Cologne runs all the animal work in my lab. And Sylvia Chung and Han Shan for the glioblastoma experiments that I showed. And the Maria Solly and David Ziegler at the Sydney Children's Hospital for the work with the rapalogs and that combination. And the clinical investigators running the trial, Anne Hamilton and Danny Rishin, and Jayesh Desai here, and Lisa Horvath in Sydney. And these are the funders of the work. Thanks for your attention.
So in your trials, in the beginning you mentioned that the cancer cells which rely more evenly for glycolysis and the mitochondria respiration. So in the drug, the concentration you put in there is messing with ANT, which is in the mitochondria.
Yes.
So in your test, one result in your test assay, the death of the cancer cells more rapidly compared to control, which is actual side. You use the opposite, that the normal cells, which rely heavily on the mitochondria, are the cancer cells.
Okay. Yeah, I see where you're coming from. So what we know, so first of all, if you mess with mitochondrial function in cancer cells, that's not good for them. They'll die. So, their reliance on mitochondrial oxidative phosphorylation for ATP has changed, but they're still absolutely dependent on it. Okay. The reason why we're seeing, I think, selectivity for tumour cells is that it's really targeting, it's affecting proliferating cells. So when we incubate our compound with growth quiescent cells, they're perfectly happy. So the cells need to be proliferating before we see this functional effect, the consequence of mitochondrial perturbation. So that's, I think, it's one of the major sources of selectivity. And the other one is that we just got lucky with the compound. It's great pharmacokinetics. It's very water soluble. It'll be largely intravascular in terms of its distribution in the body. And so, the first proliferating cells it's coming across, if you've got robust solid tumour growth will be those tumour cells. So I think that's a big part of the reason why we see the selectivity. There's also a measure of selectivity in terms of whether the tumour cells can pump this out. So drugs are pumped out of tumour cells by the multi-drug resistance associated proteins, as you would know. 2 of them can pump PenAO out, but the others can't. So, it just so happens that our rate of accumulation in tumour cells is such that they just don't efficiently pump it out. So that's working in our favour as well. So that's probably where the selectivity is rather than, we're specifically perturbing the Warburg effect. It's more of a general perturbation of mitochondrial function.
Up the back.
I'm just wondering, so it's nice that your compound has specific target to A and D.
Yes.
It wasn't clear to me the process that you took to come to conclusion that it is A and D.
Okay.
And if the process look at many other proteins, how many other off targets do you have for the compounds?
Yeah. Look, we know that A and T is not the only thing it's interacting with. There are other proteins with these closely spaced cysteine thiols. We looked at some phosphatases, for example. There's some glycolytic enzymes that it can interact with, but not terribly strongly. But what we do know, for all we know, this seems to be the functional consequence. So when we perturb A and T, this is giving us our functional consequence, our proliferation arrest and ultimately cell death. So like any small molecule, it'll be binding to other things. But in terms of the functional consequence for the cell, this appears to be what it is.
Do you have an assay for AMT function?
Yes, there's some crude ones. You can just look at mitochondrial swelling and you can look at ATP, So we can do that in the intact mitochondria, and we've obviously done those studies and we show we can perturb those things. But I think the key is that those experiments that I showed you, this was done in viable cells. So in terms of the effects on the viable cell, we were talking about mitochondrial perturbation. And then we went to the purified mitochondria to try to figure out the target. And it so happens that the target is actually the most abundant protein on the inner mitochondrial membrane, so there's a lot of target there for it.
So, since this affects proliferation, what about cancer stem cells?
We don't know. We haven't done any investigation in that area at all.
Hi, thanks for a very interesting talk. I've got a couple of questions. Firstly, with regards to the first part of your talk, where you talked about one configurations.
Yep.
There are surely more than 2 to the 5 possibilities because you're really just categorizing them. Is there any? And so the first question is there any evidence to look at more to break that down into further subcategories?
Yes, it's a good question. Sorry, you want me to answer that one first? Okay.
I'll answer everything otherwise. And the second question is, given that you're using FDG PET in your clinical trial,
yep,
and given that this decreases metabolic activity, would you worry that you are artificially decreasing the FDG the FDG uptake, so you get a false reassurance that the tumor is no longer there when there are in fact still viable cells in the tumor? And in that case, could you do something like add radiation therapy on top of that to wipe out the rest?
Okay. In terms of the first question, yeah, it's a beauty. So we did look at it. There are other ways of refining those 20 types further. But this is where we sort of got this is where it became informative. We looked at 6 configurations, then we went to the 20, and we could take that 20 to 40 or even more if we wanted to find details of the configurations in terms of the individual bond angles. And there's some rationale for doing that, but it didn't give us any more resolution. So that's why the 20 seems to be about right in terms of giving us that resolution. In terms of the next one, look, FDG PET's not perfect, but it really is, it's the best clinical indicator of an effect on tumour cell metabolism. Okay, it's commonly used, routinely used in oncology settings. So we're hoping we'll see a change. It could go either way, it doesn't really matter, but it's about seeing a change. But ultimately it's tumour size that will determine whether we see whether this compound has legs. And so that's that's really the key. At the end of the day, you want to see shrinkage of your tumour more than 20%. And that'll give you some really confidence that you're probably affecting the tumour.
Because what I was actually afraid of was false reassurance by your pre-treatment FDG applies like Christmas tree.
Yep.
And then post-treatment, it all goes down, and you can say, well, yeah this works.
Now, I know what you're right, but that's not, you're not relying on that. It's actually tumour volume So the combination. If you saw a change in FDG PET, you know, we'd still be uncertain as to the, you know, its veracity, but it's the combination.
Is the mechanism behind the kidney toxicity understood?
No, not really. What is known is that in the distal tubule, this is where metalloids are detoxified in the kidney, and so it's possible that this is where we're seeing the toxicity due to their detoxification in the kidney. Whatever it's, the concentration is just overwhelming that process. So there is that rationale, because metalloids like antimony, arsenic, and other sorts of things, this is where they're detoxified in that particular part of the kidney. So that's what we think is the case. But we really haven't, in the clinic, we haven't seen any kidney toxicity at all.
So that was just in the mice?
That was just in the mice and the rats.
So it sounds as if the patients that are recruited for the phase 1 trials are desperate. They're people that have had no responses to other treatments.
Yep.
And so sometimes from a phase 1 trial, which I know is only designed to look at toxicity parameters, you can get encouraging insights into whether those patients are potentially living longer. Have you had any encouraging results like that?
Look, we've seen some stable disease, but in not at a statistical level. Based on our preclinical data, probably where we'd like ultimately to get is pancreatic and brain, but they're typically not recruited in these sorts of trials. We have seen some nice stable disease in ovarian cancer, but you know, just sort of anecdotal, you know, just the odd patient or two. But yeah, nothing, you know, nothing that jumps out at you.
And in principle, there's no reason why you couldn't use this treatment for liquid tumours as well?
We tried that in animal models. We see no effect. It really is quite, very little effect in terms of leukemias.
Maybe a couple more questions?
Isn't actually using the panel to inhibit ANT actually inhibiting the oxidative phosphorylation?
Yes, it is.
Glycolysis.
Yeah, so one of the studies we showed, with you administer PNA, you see an increase in acid production in the cells and a decrease in oxidative phosphorylation. So it is inhibiting oxidative phosphorylation and mitochondrial oxidative phosphorylation.
In relation to the selectivity, can you just remind us or tell us about the, in the animal tox studies, what happened to, say, the lymphocyte counts? Did they get lymphopenia or not?
No, really the only toxicity, what defined the MTD in the animals was the kidney tox. We didn't see any changes in the hematological parameters. In terms of coming back to the leukemia, we think with the leukemia cells, they're actually very good at pumping this compound out. They express this sort of 2 of the 9 or 10 transporters that do pump this compound out. And we think, you know, that's why we think they're quite resistant.
Okay, one more.
It's a quick one.
Yeah.
Do you have a simple assay, or have you looked at whether you could identify at a proteomic level everything that gets hit in cells?
Look, it's possible. You could sort of a differential and alkylation approach would get you there. But it would be a huge undertaking, and we had thought, when we were trying to figure out how this was working, we thought this might be a way to go. But you know the nature of those sort of studies, it is an enormous undertaking, and at the end of the day, you still don't know what the functional consequence is. For instance, we may find this binds to actin terrifically, but does nothing to actin. You know, so we really would try to, you know, restrict our studies to trying to sense a functional consequence in the cell. And that's what led us to the mitochondria.
Okay, thanks Phil for a lot of thought.
I'm glad.